Hi, everyone. Today we have a special episode of In Rare Company podcast. It's just me and Gilbert co hosting today. We're going to be talking to someone super special in the Barth Syndrome Foundation community. Her name is Lindsay Marjoram. She is the director of research, and she is super knowledgeable about everything Elamipretide related, about drug development. So she's going to walk us through and give us a real primer on Elamipretide, and we're going to be going to school today. So get ready and hope you enjoy this episode. Cool. All right, awesome. Well, we usually start the podcast by talking about snacks. Are you eating any snacks?
No, but I did just finish boba from one of our local places.
Oh,
I
nice.
DoorDashed it earlier.
I haven't had boba in a really long time, like maybe since high school, but I feel like
Really?
it's become so much more mainstream. Like it used to be
Yeah,
like a very niche thing, and like there was one tea house we could go to get it, and now it feels like it's like
it's
everywhere.
everywhere. Yeah, it's funny. Melissa and I have a habit now of any place we travel for BSF, we try to find like the best boba place in that area. And so sometimes we'll test two or three places depending on how long we're there. But it's a good ritual for any city we've gone to. We're pretty savvy at finding boba in DC now,
I
as
bet.
well as Florida.
I feel like you should have like some kind of it should be a tab on the Barth Syndrome Foundation
That's
website.
right.
Like if you want some good reviews of boba.
That's right. And then other people can submit their recommendations for for new cities we might travel to.
Yeah.
Yeah, that was my snack today because it's very hot here in North Carolina. So it's in the 90s and like feels like 1,000,000%
Yeah.
humidity.
I'm sure. Because you're out like you live near Chapel Hill and Durham
Yeah,
and all that, right? Yeah.
yeah, yeah.
Yeah,
So
I do not envy that. That's why I'm like, I enjoy a dry heat. Colorado's good because it's
it
hot
is.
and the shade, you're like, oh, there is a temperature difference.
Yeah, it makes a huge difference. There's no relief here. It did rain this morning, and so it's actually not as bad as it was yesterday. Yesterday was miserable. And we're on the year-round school system here. So my kiddo actually started middle school yesterday, and the next 3 months are going to be like walking in jello to and from school.
Anyway, so I'm excited to kind of get like hear your take on everything because I think one of the things that like in doing the podcast, we've gotten like some good like personal stories about like how Amiprotide has affected and impacted our lives, but it'll be really cool to hear like the science side of like how it works, where it comes from, like all of that.
Yeah,
Um,
yeah.
yeah.
This was a really fun exercise for me because I hadn't really delved into all of the history. I kind of knew from Stealth's development forward, but I really got to go back into the literature and understand a little bit more about it. So I did learn because I was speaking with somebody at Stealth today because I knew there was some specific meaning behind the word elamipretide, which is really hard to pronounce. There's too
Yeah.
many vowels.
Yep.
um, I am assured that the trade name, so the name it will get once it's approved, is much easier to say.
Is it
Um,
Bengavia?
no, that was
Oh,
so
they
that was
changed
its
it.
original name. Yeah, but what it is that so the mipretide, that's Stealth's hallmark, and it stands for mitochondrial protecting peptide. Um,
Oh,
so
that's cool.
that's what the end of the word is, and they have other drugs in development that have end with the mipretide.
Retired.
Yeah.
Okay, all right. This is making
See?
sense. Okay, so yeah,
Yeah.
what's the rest of it mean?
I don't know. Katie was actually going to see if she could find out if there was any meaning to the first three letters. But yes, the ending is mitochondrial protecting peptide. So, yeah. Well, I figured I could tell you a little bit about where I come from because that kind of will shape maybe the lens through which I understand all of this. But so I'm a New Jersey native. I've been with the foundation for a little over a year and a half. I did an undergraduate degree in biology and did undergraduate research in developmental biology. So, really understanding how embryos develop to become, fully functional people, or in the case of my work, sea urchins. um, but I did that in Pennsylvania, and then immediately after graduating, I went to Vanderbilt for graduate school.
Okay.
So I was in Nashville for 6 years and did a PhD in developmental biology and moved up to frogs. And I actually studied the heart. And so I don't know if you're aware, but the heart, it's not symmetrical in your body. So when you pledge allegiance, you always put your hand over the left side of your body, and that's because your heart is on the left.
Mhm.
And there's actually a very important, very early process that tells your heart to go to the left. And so if that doesn't happen right, the heart might end up in the center. It could end up on the right side. And all the blood vessels that kind of come into the heart might not connect right.
Okay.
So that really started my early love affair with kind of the heart and cardiology and how, things really, really early on in embryonic development can impact the way the heart functions when you're born.
Hmm.
And so I studied that process in frog embryos, and then finished there and moved over to Duke. And I was a postdoctoral fellow at Duke for 5 years and switched systems. So from frogs to fish. So I was in zebrafish, which is another really old and traditional model for developmental biology,
Yeah.
but studied how the gut functions, so how your intestines kind of develop and function.
Hmm.
And then finished there. And actually, in the last year of my postdoc, had a baby. And so that was many, many moons ago now, because that baby is now 11 and in middle
Yeah.
school. And went into industry. And so I was in industry for about 8.5 years. First for a company that made diagnostics kind of similar to what a COVID test looks like,
Oh,
but for different purposes.
yeah.
And then switched over to a different kind of company called a contract research organization.
Mhm.
And so those are companies that really do experiments for other companies. So a lot of times a company like Stealth might need to do a study in mice or in rats, but they don't have their own animal colony. It's very expensive to maintain your own animal colony if you're not at a university. And so
Okay.
companies like Stealth use companies like the one I worked for to do their animal experiments. So that's where I was for the last 4.5 years, where I got to learn a lot about drug development, a lot about kind of how the FDA works, a lot about gene therapy,
Yeah.
and I also kind of ran my own lab while I was there. And then was looking for a change, and there was an opening at BSF, and it was just kind of a fortuitous introduction from another colleague. But with my love of heart development and
Yeah.
really feeling a need to be connected more to the patient community. That's something, when you work for a contract research organization, you're 2 to 3 steps removed from the person that's at the end of the, receiving end for the product. And all the clients we worked with, they all worked with patient foundations. And I was, really feeling like I was missing that. And so it was a really amazing opportunity. And this is by far the best job that I have ever had. It's truly, truly rewarding. It's challenging. Like I said, I've learned so much. I'm going to share my screen. Let's
That's
see.
really cool though. Like you're saying how you kind of happened upon the Barth Syndrome Foundation, like with like how interested you were in hearts and all of that. So yeah, I'm
Yeah.
glad that it's like such a nice fit, like professionally, like emotionally, like you're saying, feeling connected to the work. That's really
Yeah. Very much, it doesn't get more connected than this.
cool. Hmm.
And, really helps me understand, who I'm working for.
Hmm.
So I'm going to share my screen, but I'm going to have to do
Hmm.
this. Okay, so can you see my slide?
Yes.
So, like I was saying, the one of the best parts of this job is the people.
Yeah.
Both my team, we really, and Laura's not pictured here because she wasn't at the conference last year, but she's a new member of our team. But truly an amazing group to work with. And then just so many amazing community members, so many fun children to snuggle.
Yeah. Oh, I know. I
And
like. everyone to me is like a celebrity. So I'm like, oh my gosh, I recognize that person, that person. I'm like, yeah,
I know. It's just so fun. Everybody's been so welcoming and I really felt like I fit in right away. And then professionally, so on the kind of the right-hand side of the slide, there's so much, so many different researchers that I get to engage on a day-to-day basis. I don't feel far from the science, if that makes sense. Like I still feel like I really have a pulse on all the research that's going on, but it's been a really lovely way to reconnect with people from all across my professional life. So, we held Mito 101 last year, and Jessica's actually, she was a graduate student when I was a senior grad student at Vanderbilt. And so she's actually a middle school science teacher in Denver.
no way.
Yeah. So she hosted Mito 101 for us, and Virginia is a feeding therapist that we've worked with personally here, and she gave a fantastic roundtable. And then Victoria is a lactation consultant who gave a wonderful roundtable. So it's been really cool pulling people in from across the different facets of my life you know to build more research in Barth syndrome. Erica Davis is somebody who uh, she was a research assistant professor when I was a postdoc at Duke. She now has her own lab in Chicago and has helped us out a few times now. And so it's been really cool, again, to just bridge these divides and bring more people into the fold.
Mhm.
Um,
Yeah,
but
so
for
cool.
to,
Like
yeah, go
Walker
ahead.
was talking when we interviewed him, he was talking about like, just how incredible the organization is. Like how none of this really would be possible without BSF and how like connected all the like research and the families and the advocacy, like it's such a strong organization. And like I can see that from just from this slide of just like, look, this is all about the people. And there's
Yeah.
really good people and really, really smart people working on everything.
And I think that really is a testament to the families that came together 25 years ago who
Yeah.
really had the foresight to, intertwine advocacy and research and, one doesn't stand without the other.
Yeah.
And I think the advocacy component for me was really missing in my professional life. And so it's very present now.
Yeah.
But so we can hop in now if you want to Elamipretide. Um, so I know you guys have covered a lot of this already, but just to kind of review some of the basics in case anyone's tuning in that is newer, because there's always families that are kind of entering the fold who have just found out that their kiddo has Barth. So, every human has DNA in their cells, and DNA is what kind of controls how our cells ultimately behave and function. And sometimes there's changes in the DNA, and sometimes the changes don't mean anything, and sometimes they actually improve something, and sometimes they make something worse. And in the case of Barth syndrome, there are changes in the DNA sequence for a gene called tafazin. Um, and what that ends up doing is impacting the mitochondria. So if this is a human cell here, the cell is full of mitochondria. That's what a cartoon version of one looks like. I think a lot of people are familiar from their maybe high school days of hearing that the mitochondria is the powerhouse of the cell. That's kind of the stereotypical thing that people say.
Yeah.
And what it does is it makes energy. And every cell in the body, with the exception of red blood cells, has mitochondria. And some cell types have more mitochondria. So you can think about organs like the heart that do a lot of constant work. They have a lot more mitochondria than maybe a blood cell, a different, type of blood cell would. And so in somebody who does not have Barth, the mitochondria have all these little nooks and crannies. And it's actually formed because there's two sets of membranes. So you can almost think of like two fences that pack a lot of small proteins inside the mitochondria in order to make the energy. And all of the energy making machinery is shown here, and it's not important what they are for this purpose, but they kind of do all their work in these little nooks and crannies. So it's really important that the mitochondria has all these tiny and tight folds because that's what puts all the machinery together in order to do the work. In Barth syndrome, what ends up happening is that when this gene isn't working right, there's a pileup of a special fat called cardiolipin. And cardiolipin lives on this inner membrane. And the pileup happens because the very mature form of cardiolipin doesn't get made. So, kind of an immature form, which you'll hear referred to as MLCL or monolysocardiolipin, it's a real mouthful. Um, it causes a pileup of that, and the mitochondria become misshapen. So you can see this one's swollen. It's like bigger, and the
Yeah.
folds aren't as tight. And so what happens is it puts the machinery further apart from each other, and they just don't make energy as efficiently. And that's where everything kind of starts from. So if you can't make enough energy, then all of these other things start to happen. But Elamipretide, they found, is kind of like a molecular glue, and it pulls all the folds closer together. So
Okay.
it's just helping the mitochondria have a more normal shape so that all the pieces of the machinery that make energy can work together again in a more efficient way.
Okay.
Um, so that's kind of an overview of how that works. I don't know if you have any questions from
Yeah.
that.
Yeah, I think like when our cardiologist explained kind of this to us, like the thing that I took away in my mind was like, okay, it's almost as if his mitochondria are having a hard time like communicating across the mitochondria because, and she mentioned, and maybe we'll get into that, like ATP.
Yeah,
Like,
so that is the energy. That's like the currency
okay.
that the mitochondria make so that the cells can do their other jobs, right? So ATP is the payload from all of this machinery here. And you need, it's kind of clipped off right there, but you need ATP. It's like paying tolls so that the cell will do the rest of its job. And if you can't do that, and the mitochondria looks more like this, they start to make kind of unhealthy byproducts, something called reactive oxygen species that makes the mitochondria unhappy, which then spills stuff into the bigger part of the cell and makes the cell unhappy. And they just don't do work. They don't do the work that they need to as much as they should.
Okay. And so like the Elamipretide, like you said, it's kind of like a glue, so it kind of like bridges that gap
Yeah.
between the
It's just kind of pulling everything together so that this turns back into this, right? So it's turning more into, a bean shaped object where, again, it has all those tight folds because that's where all the work is getting done.
got it. Okay, cool. Yeah, no, this is so that what a like beautiful graphic too. That's so, did you make this?
I did. I made it in BioRender. So like these are from, and this is actually from a publication from the person who originally discovered the peptide that is Elamipretide. But
Cool.
yeah, BioRender is a great program for making scientific graphics.
Yeah. Yeah, this is awesome. I love it.
um, so I thought I'd give a little history. Well, first I thought we could maybe talk about how do drugs get made?
Yes.
Because I think that's that leads us to where we are today, and you can't get to there without talking about this.
Yeah.
So I'm going to talk about it more broadly here, and then I'll go into specifics in a minute. But it really all starts with somebody having an idea, which sounds simple or basic, but that really is where all of it begins. So somebody has an idea about a disease or a process, and they think, I want to explore that. And so that's where discovery research comes in. It's not exclusively performed at universities, but a lot of discovery research is performed at colleges and university labs.
Mhm.
And so that's what we sometimes call basic science, because it's not necessarily aimed at identifying a drug, though sometimes it is. Sometimes it's just to understand a process. Um, and so discovery research, can take any amount of time. Sometimes it goes fast, sometimes it goes slow. Sometimes you set out to find one thing and you end up finding out something completely different.
Mhm.
Um, But when you're on the drug development pathway, if you're moving towards understanding how to make a drug, typically in your discovery research, you've found something that seems promising. And so before you move into what's called a preclinical research program, you might talk to the FDA and just say, hey, I've got this idea for a drug. I have this, preliminary data. Um, and they'll give you feedback on how you should do the next steps so that that can turn into something that helps you run a clinical trial. Because there's a lot of checks and balances along the way before you can actually put something into a human.
Mhm.
So you might have, not everybody has an interact meeting, but often people do because they don't want to find out that they've done all this preclinical research and that they missed important things and
Yeah.
have to go back
Yeah.
because then that wastes time. So like I said, FDA might help shape this, but preclinical research, I've put these little graphics here, might be done in a mouse. It might be done in cells in a dish,
Mhm.
and it might go as far as even into monkeys or pigs, so a bigger animal,
Mhm.
to better understand how it's going to work when you put it in a human.
Yeah.
And a lot of the purpose of these, there's kind of two main things that you need to understand from this research. The first is, does the drug work? So is it fixing the problem you think it's supposed to fix? And then is it safe? And that's a big one, right? Because you don't want to put anything into a human that might not be safe. And you want to know it early, not later. So after you've kind of done all of that work, and that work can take a couple years, that's, it's not a short process. You then show all of that data to FDA in something called an IND. So you first have what's called a pre-IND meeting. IND stands for investigational new drug. And then once they've kind of signed off and they say, okay, go ahead, submit it. Um, they will give you the green light to do clinical trials. So maybe first in human, or, if you've heard of phase one, phase two, phase three, clinical trials. Or they might say, hey, you actually need to get some more data. We're not sure about X, Y, or Z. Could you please go do these experiments? You'll go back and complete them, and then you go back to the agency. So then the other thing that when you go to FDA, you also tell them your plans for how you're going to run your clinical trials. How many people are you going to enroll? How long will the study be run? Who will be enrolled? Are you going to exclude people who have a certain condition or not? So, in the case of Elamipretide, in that trial, people with heart transplants were not included in the trials. And that's often a really challenging point. Heart transplant recipients take a lot of medications. And so they're sometimes more challenging to include in a trial because you have so many drug interactions that you have to factor in on top of just looking at how the drug itself works.
Yeah. Well, and then that must have been a particular challenge for our community because a lot of people have had heart transplants. So
Yeah.
then how do you even find enough people within that small population to
Yes.
be in the clinical trial? Yeah. Huh.
But what's really amazing about this community is that I want to say the trial was fully enrolled in 3 months. So they did a single site. So it was held at Johns Hopkins. And, they surveyed all the people who could participate. And again, it was only for 12 and older, couldn't have a heart transplant. There were a few other, what they call inclusion and exclusion criteria. Um, and they had 16 people that were eligible and 12 that enrolled. So that's an incredibly high success rate for enrolling, especially for the timing. It was very fast.
Yeah, that's
So,
incredible. When you put it
yeah.
like in, yeah, because I'm like, I didn't know any of that, but that context, that is really amazing.
So the phase one, two, and three, those are the names of the different clinical research studies. Phase one often is a study that you perform to understand what dose of the medicine is the best dose to use. But sometimes, especially in ultra rare diseases like Barth, you kind of just hop in. And at this point, Stealth had already done some other clinical studies, so they already kind of had an idea of the right dose to use. So they hopped in straight to understanding if it was efficacious, meaning did it work? And then after you do those studies, usually the studies, sometimes they get bigger as you go from phase 1 to phase 2 to phase 3. But in the case of an ultra rare disease like ours, um, there aren't more patients or a bigger population to enroll. So in the case of this, they actually took the trial out longer and longer with people on Elamipretide and compared it to what's called a natural history population. So a group of people with Barth syndrome that have not taken Elamipretide, so that you can see if there's improvements over time. But when you finish all of that, you then submit what's called a new drug application to the FDA. And the FDA has to accept this in order to review it. And the standard timeline for this is about a year, although if you get priority review, it can be something like 6 to 9 months. And then at the end, you hope that the drug gets approved. Now, if we look at Elamipretide,
Mm-hmm.
I've kind of outlined some of the timing of this. So the discovery research was done through a collaboration between Dr. Hazel Zetto and Dr. Peter Schiller. Hazel was at Cornell, and Dr. Schiller is from Montreal. And together they identified, they had the goal where they knew that when mitochondria weren't healthy, that it really contributed to things going, south pretty quickly. And so they distinctly set out to find peptides, which are small proteins, that could get not only into a cell, but then from the cell straight into mitochondria to help fix them. So they developed what's called a library. So they found many of these different compounds that could get into cells, get into mitochondria, and help them feel better. And so from there, Dr. Zetto started Stealth Biotherapeutics. And in 2014, BSF actually approached Stealth and had seen a lot of the work on what was originally called SS31 and is now called Elamipretide. And SS stands for Zetto Schiller. And asked Stealth if they would consider looking at its use in Barth syndrome. And so from 2014 to 2018, they did a bunch of preclinical work. And then they had a lot of IND discussions with FDA and got permission to run what's now called the TASPower trials. And those were run from 2018 to 2021.
Okay.
But you can see here, I've put a little ellipsis because we're not quite done with the process yet. So, from 2021 on, again, you guys have probably already chatted about a lot of this, but it hasn't been a smooth road. So after the clinical trials were done, the data indicated that Elamipretide was helping in Barth, but it wasn't what we would call clean. And that's really to be expected when you're doing a study on only 12 people in a disease that's a bit heterogeneous, meaning every person's symptoms are a little different.
Yeah.
And so there were a couple issues where FDA really couldn't decide which division needed to review it. So there were 4 different divisions that Elamipretide went to before it settled at cardiovascular and renal, so heart and kidney. But in 2021, FDA refused to file the new drug application. And so this is where BSF and Stealth really had to work hard together to advocate to both FDA and to Congress that they should accept this for review. Mm-hmm. And so it was really the community coming together and and lots of continuing conversations with FDA that happened in 2022 and 2023, culminating with a petition submitted directly to FDA by Kate McCurdy with nearly 20,000 signatures asking for FDA to accept Elamipretide for review. So in early 2024, the NDA, or the new drug application, was accepted, but given standard review, which is a little unusual given the case for Barth syndrome and needing a drug that's helpful for the disease.
And did it already
Um,
have like orphan status?
yeah, we had all the statuses that you should have for, getting priority review. So they did get assigned priority review on the back end, but it didn't change the timeline.
Huh.
We had an advisory committee meeting in October of 2024, and that's a special committee that is assembled by FDA, but
Mhm.
all the members, you can think of them like a jury.
Yeah.
They're all independent from FDA, and they're independent from Stealth, and they're independent from Barth Syndrome Foundation. So they're what people would call non-conflicted,
Mhm.
but they're experts in things like skeletal muscular diseases or mitochondria or orphan drugs or ultra rare diseases. And that committee heard all of the data from Stealth. They heard all of FDA's analyses and concerns. And then they also, for 1 hour, heard from many members of our community, both patients, family members, as well as clinicians who have used this in younger infants, just like Gilby.
Mhm.
And they voted at the end. This committee voted 10-6 that elamipretide was efficacious, meaning it works to treat Barth syndrome. The vote probably would have been even higher had the original question been, is should elamipretide be approved? There were a few people voting who said had the question been different, their vote would have been different. Um, and so our original decision date, which is called PDUFA, which is another acronym, lots of acronyms at FDA. So that's the Prescription Drug User Fee Act date, was in January, and then it got bumped to April of 2025. And then that date was missed. And unfortunately, in May of 2025, a CRL, which is a complete response letter, was issued by FDA. And that's what they issue when they're not going to approve a drug at that time. And they also said that the path forward would not include infants. And so that's put us into a little bit of uncharted territory, and not just us as in Stealth or us as in Barth Syndrome Foundation, but really as a whole, this is a little bit unprecedented in drug development. But we know that in June, Stealth had what's called a Type A meeting with FDA, and that's where you kind of review what was in the CRL and talk about what the steps forward might be. And that it was a very positive meeting, and that some of the things that were mentioned in the complete response letter were addressed. And so Stealth was encouraged to ask for reconsideration. So around July 1st, the week of July 1st, they submitted a formal request for reconsideration, and the agency has about 30 days to respond. So we know that around the beginning of August, Stealth should be hearing from FDA. But as to what the next steps will be, FDA's answer is really what's going to shape what happens next.
Yeah. Yeah. This is all like, again, such a beautiful graphic. And your explanation is like so nice to kind of like just parse it down into those steps. So,
Yeah,
do
happy
you
to.
know, like something that like I've wondered about in all of this is like the movement around the different divisions. Like, do you know why like we were moved around so many times or
I
how that
think
works?
they really had a hard time figuring out what kind of a disease that Barth is. There used to be a metabolic division, and I think that got either dismantled. And so part of it was like structural changes within FDA. And so that's why, like, I think it was about finding the right fit. So finding the reviewers who were really going to understand Barth syndrome the best. And, when you look at what's most severe in Barth syndrome, it's the heart and the immune system, right? So,
Mhm.
folks with Barth often have heart issues and they can be life threatening, or
Mhm.
maybe they have issues with their neutrophils and they don't fight infection very well.
Mhm.
And, so they're subject to things like sepsis. Um, and so, but I think overwhelmingly the heart issues are what come to the top of the pile when you think about the severity of Barth. And so I think that's why ultimately it ended up at, cardiorenal
Yeah.
division.
Okay, that makes sense. And then in terms of like the changes with, or like, with having the NDA accepted, but then it being assigned priority review later, Gilbert, but that not changing the review time, like is there precedent for that with other rare disease communities and other drugs developed? Like is there reasoning behind why things might take longer than expected?
I don't know the answer to that, to be honest. I think it was because it was assigned priority review on the back end. That was a little unusual that it wasn't assigned right out front. And because the dates were kind of already in place, um, it might have just been too tricky to start trying to change the dates, especially with knowing that there was probably, we'd requested an AdCom like as BSF, and so fitting that in, and maybe it was just too challenging to try to shift all the dates back and still get an AdCom in, which was really important. It is the only way. So an AdCom or advisory committee meeting is the only way that the patient's voice is required to be listened to in a drug review process. And again, that open public hearing, that 1 hour in the middle of the advisory committee meeting is where the patient voice can be heard.
Mhm.
And so it might just have been a little too tricky. It's really hard because, right, FDA is very big, and
Yeah.
so it might have been hard to change all those dates and accommodate an AdCom meeting.
Okay. Um, yeah, when you talk about like the question in the AdCom meeting, like how you said, like if there might have been a different question, how do they determine like what the questions are that they're considering?
FDA is the one that determines that. And so they had a set list of questions, and they kind of start the morning off with, this is why we're here today. We're here to answer these questions, or to see if, the answer to these questions is yes or no. And so FDA is the one that determines it, but it is an impartial third party, the advisory committee, that gives the answer to it. And like I said, the questions ended up focusing on, you know, does elamipretide work for the treatment of Barth syndrome? And sometimes when you're in ultra rare diseases and the trials aren't perfect and the data are a little messy, it's really hard to get to a firm yes. But
Hmm.
as long as the drug is safe, which, elamipretide has many, many years of human safety data,
Yeah.
people are likely to approve it because it's safe, and we can basically, you start collecting additional data on the back end, and that is the plan. If FDA reconsiders,
Hmm.
Stealth will perform what's called a post-marketing study or post-approval study,
Hmm.
and it will likely be performed outside the US because the drug would be approved here,
Yeah.
if it were approved here, and they'll look at it for longer
Hmm.
in, you know, in a probably a larger group of affected individuals.
Yeah. Um, Oh, I think I see about that. Oh, yeah. We like watched some of I mean, I think Andy watched like all 8 hours. I watched like bits and pieces of it. But I mean, it really was very interesting listening to kind of the deliberation and the way that like the different
Yeah.
scientists were talking about what their thoughts were. And it just really seemed like there were so many that were like, look, I I work with, families and patients with rare disease, and I want to be able to say yes to this, but I have these concerns. And so it was like not for a lack of like, yeah, it almost feels like there's kind of the sticking point in this back and forth with the FDA and with rare diseases and patient families, where it's like people want to be able to say yes, but then there's also this like scientific standard that has to be met that feels tricky for rare disease
Yeah.
communities, because yeah, I mean
Yeah.
anybody who works with any type of rare disease or cancers or anything where it's like you're looking for treatments and you don't want to be saying no to people, but then there's like this scientific standard. It just, I don't know, it
It's
almost,
really tricky.
yeah.
Yeah. I think Barth syndrome is really a litmus test,
Hmm.
especially for kind of the new configuration of FDA following the election. There's new people kind of at the top of FDA for how rare and specifically ultra rare indications should be handled, because I think this is a prime example where you can't apply the same rigid rules, that you might use in a clinical trial that had 15,000 participants to a trial that had 12.
Yeah.
And it's not because we couldn't enroll enough. There just aren't enough people to enroll.
Yeah.
And so, I hope there is. So at FDA, I believe it was like middle or late last year, they formed what's called the Rare Disease Innovation Hub. And it's
Oh,
not a part yet of the agency that makes decisions, but it is there to help form new guidelines around rare disease. It's still
mhm.
very early in its kind of formation. But I'm hopeful that, that the Rare Disease Innovation Hub will really help conditions like ours
Mhm.
succeed in future drug trials.
Yeah. Yeah. Because it seems like, as our technology and our ability to diagnose and figure out, I mean, like there's more rare diseases are going to be figured out. Like people are
Yeah,
going to have more research happen, and to be able to have like a process that's a little bit more tailored to like what communities that have such small populations need would be nice, because yeah, it does. It just feels like the standard that's being applied just doesn't totally make sense for
doesn't
it's like
quite work.
yeah, it's like putting on like trying to fit into some kind of clothing or something that doesn't quite fit, like something that's like too large
Yeah.
or too small, and you're like, this is not
Right.
this doesn't make sense.
Doesn't quite work.
Yeah.
Yeah, and I think, there's a lot of um efforts being placed into something that's called a surrogate endpoint.
Mhm.
So that's something that is basically an indicator that a drug is working without having to wait maybe the long amount of time for some other process that would be considered clinical
Mhm.
improvement. So, you know, something like improving your heart function might not happen really quickly, but if you had a biomarker for showing that it's trending in the right direction, that can help you get the drug approved faster. That's
Yeah.
something new that there's a lot of research that has to go into kind of validating that biomarker before it can be used in a trial.
Yeah. Yeah. Well, that makes me think about like with just because Barth syndrome presents so differently in each individual,
Yeah.
like for Gilbert, like Elamipretide certainly has stabilized his heart function. The biggest like data point we have though is his ability to eat and like how much weight he was able to gain and how he was, able to like go from having an NG tube. He got his NG tube out within a month of starting Elamipretide.
Oh,
He was able
wow.
to do full feeds on his own. And then
Yeah.
once he got home, transitioned to breastfeeding. So
Yeah.
like, and his weight gain, he basically caught up to, you know, he's at like the 40th percentile now. And before he
That's
was at the
incredible.
0.2 percentile, like he wasn't even on the growth chart.
Right.
So like, yeah, that kind of makes me think like, yeah, there's got to be some other data that you can look at.
And it's tough because if those data points are different for each kiddo, especially like in the little ones,
Yeah.
because it could be, meeting developmental milestones,
Yeah. Yeah. Yeah.
crawling on time or being able to sit up, because one of the tricky things with little ones that have Barth is their muscles are what we would call, they have like hypotonia. Their muscles are soft, they're a little floppy. And so it's harder to use. Again, you have to make the ATP, you have to make the energy to make those muscles contract.
Mhm.
And so you could look at something like sitting up on time or closer
Yeah.
to on time. But for each baby, it might be different. And so there are other things that people are advocating for, like kind of composite ways of so it's a scoring rubric for a couple different things. That way, if one baby met this milestone early and another baby met this milestone, that again, if you score above a certain number, it's still indicating that the drug is working, even if each baby is improving in a different way.
Yeah. Yeah, that would be nice because it does seem like, in the community and getting to interact with like other families that have babies who've been on Elamipretide, everybody's story is a little bit different, but there are certainly indicators that Elamipretide is doing something really good and it is
Right.
helping stabilize mitochondrial function because you can see it in the way that, your child is developing. Like, yeah, I mean, Gilbert is sitting up independently at 6 months. Is he
That's
rolling?
huge.
No, because he's refusing. I mean, he can roll from out of tummy time.
Oh yeah.
He still won't like roll from back to tummy. We're like, dude, you sit up. Like, what are you doing?
I don't know. I mean, my non-Barth kiddo also did not do that for until like 10 months old. And
Okay,
he
so yeah, I'm
only
like,
rolled out of tummy time because he hated tummy time.
yeah. His nurse, like we have a nurse that comes to check in like every couple of weeks, like that was helping us when we like first came home. And she was like, maybe he just doesn't want to roll over. And she's like, and like, I don't want to be rude, but like, he's a little fat. And I was like,
It could be harder.
and I just looked at him, I was like, Gilbert, are you a little fat? I was like, but that's
No,
good.
he looks wonderful.
It was just so funny that she was like, I don't want to be rude. I was like, no, it made remind me of that like SNL baby Spanx. Did your baby need baby Spanx? But anyway, yeah, yeah, it does seem like, there are sounds like there's lots of good ideas for ways that we can move in the direction of being able to compile data in a meaningful way for rare communities. It's just like seems like not it's not quite there yet.
It's almost like you have to build a couple different planes at the same time and fly them at the same time so that you can have it all come together. Because sometimes what's happening is, in the case of stealth, like they ran their trial, and I think one of the things they learned on the back end was just that the drug doesn't work as fast as what they thought based on mouse studies. And that's common. Mice aren't humans. And so sometimes you have to wait till you get into a human to really understand how the drug's going to work. And so, for when they do, if they get approved and they can do their post approval study, that study will run much longer because they know it takes longer.
Yeah. Oh, can you talk a little bit about the mice too? Because I don't know, like I did see one picture on Facebook. There was one of the members of the community who was like, Barth man, meet Barth mouse. And I was like, oh, that's so cool. But yeah.
Yes, I
And
think
also
that's
like I've
John in that picture. So, it's interesting because there's now several Barth mouse models. So the original work for Elamipretide was done in what's called the knockdown model. That was the very first model that scientists were able to make. It turned out not to be trivial to make a Barth mouse. And so the first model, what they do is basically it's a normal mouse until you give it a special antibiotic called doxycycline. And when you give it that, based on the way that the mouse has been engineered, then tafazin goes away. So they actually give the doxycycline in the mouse's food, and it's like turning the volume down on how much tafazin the mouse has. So that was the only model for a long time, and that's where the work for Elamipretide was done. Since then, literally like in the last maybe 5 to 7 years, there's now some really elegant mouse models that are closer to what a Barth human experiences, including the very recent ones from both Dr. Peter Relling's lab and Dr. Simon Conway. And they actually made mice that are based on mutations that humans with Barth actually have. So they used CRISPR to change the DNA of the mouse and make it look like a specific Barth patient. But those mice came much later than when Elamipretides. Remember, when we were looking at kind of the
yeah.
graphic for drug development that came way after, the preclinical work that was done in 2014.
So is that the knockdown mice? That is the image that everyone talks
Yes,
about where you can see the three. And those are images of the mitochondria, right? Not
they are.
Oh,
So
okay.
it's a special kind of image called electron microscopy, that allows you to actually see parts of a cell. So it's like a super microscope that really lets you actually see into a cell with that kind of resolution where you can really see the mitochondrial structure. Yeah, those pictures are really neat.
Yeah. Well, and that's interesting to know that that the difference between the types of engineering of the mice. I wouldn't have known that.
Yeah, there's a whole evolution that's happened. And, just because technology has gotten better in terms of how we make models of human disease. And so, I'm sure there will be, a continued evolution in how people model disease in different animals. It's now possible to use CRISPR to make human disease models in pigs, which wouldn't have been possible kind of 10 years ago, maybe a little bit more than 10. But to have a large animal model is something that could be really beneficial down the road, because it's always good, to do experiments in mice, but a mouse is very tiny. It only weighs 20 or 25 grams, and humans are measured in kilograms, so much bigger. And so sometimes not everything translates from mouse to human. And so doing an experiment in an animal like a pig or a monkey is a better proxy for how that drug's going to act in a human, so that your clinical trial goes better, because you understood the drug better early on.
Yeah, that makes sense. And that's interesting. I didn't know pigs were used because,
Yeah.
but I
Pigs
mean,
are
yeah.
really common in heart disease research because their hearts are very similar to ours. So if you like, stents that are used, all of the stent research was done in pigs.
Oh, wow. Oh,
Yeah.
fascinating. So then I guess this would probably be a good segue to talk about what kind of future research or things are kind of in the pipeline for Barth, and if there's
Yeah.
because one of his nurses always talks about oh man, it would be nice if you didn't have to give him a daily shot, what if it was once a week or once a month, or the research we saw that came out about what there was a different drug or something
The
like
mitoconic acid
yes,
5, that
yeah.
one. Yeah. Yeah, the tricky thing with elamipretide is that, the way that that peptide works, it can't be given by mouth because it would get kind of torn apart in the stomach. So it wouldn't be functional once it left your stomach. That's why it has to be given as an injection, because it bypasses kind of your stomach acids that might break it down or make it not function. But I don't know if Stealth is working on, different formulations. That's probably something they would look at doing after they kind of get across the finish line, hopefully. Um, but yeah, I do think, future drugs being developed for Barth, whenever I talk to scientists, I do always stress that, it would be great if it could be an oral pill, something that somebody could swallow, because a lot of our community members use Neupogen to boost their neutrophils, and that's an injection. And so it's hard when you have to do a lot of injections. It can make it harder to do it every day if you need to do that. And, a lot of our community members have G tubes. And so that kind of starts shrinking the real estate where you can put the injection. Um, so yeah, in the pipeline, there's a lot of really exciting research. We've announced some of the folks that have been funded for 2024. So 2024, 2025. I always get confused what year we say because it's 2025. But there's a lot of folks working on gene therapy for Barth syndrome. So that's actually helping correct kind of the underlying issue, which is tafazin. So, in people with Barth, for whatever reason, the mutation that's happened makes tafazin not function. Sometimes it's made, but it's still not doing its job. Sometimes it's not even made. And so you can provide a special virus that gives the protein back to the cells that are missing it. And there's several labs working on that. And BSF has funded one of the labs at Boston Children's Hospital who's been working on this for the last 3 years. There's another lab working on what's called enzyme replacement therapy. And so tafazin is an enzyme. So it's a protein that helps change one thing to another. So it's a very active thing. And so this lab is at Tufts University, and they're trying to make the tafazim protein that you can then just give. So again, it's giving it back to the cells that need it. Um, and lots of folks are also looking at different types of drugs that might help muscles function better. So I think there's a lot of promising stuff in the pipeline. And I think, we've learned a lot from going through this whole process with Elamipretide. I think we know that for future trials, we might need to start looking for surrogate endpoints. We might need to start looking for biomarkers, things that will help. We always joke like we would love like a menu. And so if you know how your drug works, then from the menu, you select these outcomes or endpoints for your trial to, again, help for the greatest chances of understanding does the drug work or not.
Yeah. Well, and like one of the questions I've gotten from like family members and stuff is like, well, when they hear about families like Hope's family who like she doesn't have Barth syndrome, but she can still use Elamipretide. Are there I think I've read about like dry age macular
Dry
degeneration.
AMD,
Yes,
age-related
yeah.
macular degeneration. It's a
Yeah, so there's
mouthful.
that, but like, in terms of like the broader mitochondrial disease community, like is Elamipretide something that um has there been research on other mito conditions and its effectiveness or.
Yeah, they have other trials going on right now, and I think Stealth has learned a lot about which type of mitochondrial conditions work best with Elamipretide because there's mitochondrial diseases that are caused by mutations in your DNA that lives in your nucleus. So that's kind of the boss of your cell for every cell. But within mitochondria, they actually have their own DNA. And so there's another set of mitochondrial disorders that are caused because the mitochondrial DNA are mutated.
Which
And
one is
what
Barth syndrome?
Barth is what we call a nuclear mutation. So it's in the DNA in your nucleus.
Okay.
It just impacts the mitochondria.
Mhm.
But the ones that are mitochondrial DNA mutations, those don't seem to respond to Elamipretide as well. And part of it, again, is because those conditions tend to be even more heterogeneous, like very different patient to patient. And
Mhm.
so, again, they're rare and it's hard to get a patient population that all look the same.
Yeah.
So it makes it's just a higher bar that you would have to clear to try and show that the drug works.
Yeah. And I don't know. I just feel like we really need like just some changes in terms of like how we regulate and figure stuff out. I mean, I understand the point behind the FDA because like,
Oh, yeah.
you don't want to have things that are
Not
unsafe
safe.
in
Yeah.
people, right? And like,
And
I
we have plenty of examples throughout history of things that have
yeah,
been approved that shouldn't have, you
exactly.
know. But I think that's one of the compelling things about Elamipretide, though, is that the safety data are incredibly clean. And that wasn't, for anyone who was either present for the AdCom or who sat through the 9+ hours of the recording, safety was never a concern. It was never
Yeah.
discussed, which is great. You don't want safety concerns.
Yeah.
We're seeing some of that play out right now with some of the gene therapies that have been recently approved that, again, as you dose more and more people, some safety stuff might start to come out of the woodwork. And this, again, is more in the case of gene therapies, where,
Mhm.
again, the trials are very small usually. But, if you go from 100 patients to 1,000 patients, something might start to creep out as a pattern that you didn't see in the first 100.
Yeah.
So, safety is super, super important. And I don't think anything should change there. But I do think the kind of way we view or measure efficacy in an ultra rare population needs to be different than what we would apply for something like diabetes or heart disease, heart failure.
Mhm.
Um,
Yeah. Well, and it's interesting because when I was reading the study, like, we've cited the statistic, like 97% of the time when you have an AdCom and you
right.
get a positive vote, then the drug gets approved. And I was reading the citation for that, and it was just interesting because it was like sometimes advisory committees say no and the FDA approves them anyway.
Yeah.
It's like, what?
Yeah. So
It's
it
like,
is,
wait a minute.
that, which is why there's not perfect overlap of the AdCom and the FDA, and the FDA doesn't have to follow what the AdCom says, but they do have to take it into account. And I think that's been a real pillar of our advocacy work
Yeah.
as a foundation, is to make sure that they did take the AdCom into an account, that was featured, into their decision making, because it's hard to understand with a favorable outcome from the AdCom and no safety issues why it wasn't, why the decision wasn't an approval.
Yeah. Yeah. It is
Yeah.
confusing. But well, I have one last question because we've our focus has mainly been like on, Barth syndrome, Elamipretide, like for the first like couple of episodes here. But knowing that like just in becoming a member of the rare disease community, like by proxy with Gilbert, like I've just like discovered so many things, like so many more like diseases and people, and it's like everybody's got Instagram accounts and everybody's got like advocacy stuff they're working on. So, and I've learned about a few rare diseases, like an ultra rare ones where, there isn't even treatment yet. They're
Yeah.
still just building awareness. So like for those communities, like what does the process look like? Because
Yeah.
Barth Syndrome Foundation has been around for a while because of the efforts, of like Shelley and Kate and finding
Right.
each other so early on. But like for a newer um, rare disease that doesn't have drugs in development, like what do the steps look like?
Yeah, that's a great question. And really the first part is, find your community, right? Because there's strength in numbers. And so finding a critical mass of affected individuals and their parents helps you kind of pull together honestly some of the capital that you might need to help greenlight research. So if it's an area that's new and nobody's been studying it yet, and sometimes that's the case, sometimes people have been studying it but kind of in a different context, and so the families need to find the scientists that have been doing that work, and that can be really hard, right? If you're not a scientist and you have to go digging into PubMed and read these really dense papers, it can be really hard to find the people that might be working on it. And that's where finding a lot of early or young foundations will hire like a consultant who is a scientist to help them bridge that gap. So to help them find the scientists who are either doing work in that area or work that's really closely related. And then if you have money to fund grants, that's how you can help attract people to come study your specific disease and come up with, novel ideas for treatment. But it really all starts with, affected folks and their families finding each other, and then finding their kind of counterparts or their comrades in the scientific community, which again it's not trivial.
Yeah, yeah. Okay, that's interesting. I know, because I'm like, we are coming in this late in the game, I'm like, and so
Yeah,
it's
but
yeah.
because of BSF, that's why Elamipretide even came to be for
Mhm.
Barth, right? They approached stealth. And that I think especially for ultra rare, where, you know, sometimes the math is tough for the biotech companies, right? Because if it's a really tiny patient population, the biotech company is going to invest many, many millions to develop the drug, and they may not make a profit on the back end, which means they can't pay back their investors. And so, it's tricky, and that's why we have incentives in Congress, like the pediatric priority review voucher, to help get folks into research for ultra rare. Yeah. And that's another point of advocacy for BSF, is that pediatric priority review voucher. It's fairly popular on both sides of the aisle in Congress, but it lapsed in December. It needed to be renewed. It was a part of a larger bill, and there were some other unpopular things. And so it did not get approved in December. And so right now, stealth is grandfathered in. They have, and I can explain what the PRV is, but basically it's a golden ticket. So it says if you meet all these criteria about being an orphan indication, rare pediatric population, all of these things, you are eligible for what's called a pediatric priority review voucher. And so what it means is that if your drug is approved at the end of this, FDA will give you the PRV, and you can either use it to skip the line for your next drug that you develop, or what a lot of small biotechs do is they sell the PRV to a big company like Merck or GSK, and that's how they recoup all of the money that they put in for R&D. So it's how it kind of makes it make financial sense.
It's a Disney Fast Pass.
It is a Disney Fast Pass. Yeah, it's a Fast Pass but it's like a Fast Pass you could also sell and then, get to go to Disney many times after.
Yeah.
Yeah.
Okay. So, but that legislation, they did
That
not.
expired, so it sunsetted in December, but Stealth already had it assigned, so they're grandfathered in. So if their drug gets approved, they can still receive the PRV. But
But
we
that would be
do,
something like we should maybe turn some, once we get Elamipretide approved,
yes,
if
we need
people
to
are,
go back to, so it was a big point when we went to DC in February for Rare Disease Week. That was one of our asks, was for reauthorization of the pediatric priority review voucher.
cool. Well, this will be good to like again, I mean, like, in February, I was like I would like kind of look at BSF stuff from afar because I was
Yeah.
just still like
In
in
the
the
in the
trauma being,
weeds.
being in the
Yeah.
ICU. So I was like, I can't, but I
In the
was like,
trenches,
oh,
maybe more than
what?
the weeds.
Yes. I was like,
Yeah.
what is Rare Disease Week? Like, so I was just
Yeah.
like starting to learn about it. So
Like, I have a very sick baby right now. Yeah.
like,
Yeah.
okay,
Rare
good. So
Disease
I
Week
know
is
about
great. And the cool thing is, though, in the time between now and then, you've already made so many contacts with different elected officials in Colorado. And so you're already going to have a relationship going into these meetings, and you're not starting from, ground zero asking for things. You can say, hey, as we've discussed, let me update you on this. Also, relevant to this, we need the PRV.
it. Hmm.
Um, that's what helps future drugs get approved.
Well, and this is good to know too for like people who have just now kind of joined like our family because of
Yeah.
Gilbert, like join that journey, like the journey of being like, okay, this is the next thing that like you can start calling about and that
Yes.
we start advocating for because
Yes.
yeah, I mean like I didn't know about any of this and now I do and like
Yeah.
now all these other people do too.
Yep. No, it's huge. And yeah, there's still lots of things in rare and ultra rare that come together to help support kind of the full ecosystem, right? Because you can have a company that's interested in doing the work, but if they can't get the money to do the work or they can't get reimbursed on the back end, then they might choose not to invest in Barth. They might go for something that's bigger. And so we want them to come invest in us. And so we need all of the kind of train tracks to be in place to allow that to happen. So yeah, the pediatric priority review voucher is a big one and certainly something that unless it gets passed in the interim, we need to keep advocating for. Like I said, the good thing is it is very popular.
Yeah.
So it's just a matter of kind of where does it fit in the legislative process to get it over the finish line again.
Yeah. Yeah. Well, and it's like, I don't know how it could not be popular because
It costs no money.
yeah, well, and rare disease doesn't pick and choose who it affects. So it's like
No.
across the board, across the aisle, all people
Yep.
the statistic we talk about a lot, like everybody knows somebody with a rare disease because
Yep.
when you have a rare disease population of 30 million people in the US,
Right.
like
Together, collectively, it's not
yeah,
rare. Each
yeah,
one in itself is rare. And then you have ultra rare, where it's even less frequent, but even still, all the ultra rares combined end up not being
yeah,
ultra rare.
yeah.
Um,
I know.
yeah.
Well, and into I was thinking about this the other day. Like someone was asking about yeah, like how many birth patients and like this, and I was like, to me right now, it seems like my whole world, everyone I know has Barth syndrome, but that's because this is like what I do all the time is talk about it and advocate, and I'm in the Facebook groups, and I'm
Yeah.
but when I think about it, I was like, oh wow, yeah, 100 people or so is really not that many.
Not it, yeah, in the US. And
Yeah.
globally, we know of about 300. That number is far higher. It's just that the means to genetically test and identify the cases, and then there's language barriers. So, the number is higher than 300. Is it 3,000? Who knows? Like it, it's unclear, but there's many countries right now that we're not in contact with, or haven't heard from. It's one of the things that Melissa and I recently have seen a flurry of papers in countries where we're really not aware of many or any affected individuals, where they're talking about cases. And so Melissa and I have actually been reaching out to the authors and trying to set up calls because we want them to make sure that their affected individuals know about BSF. But it is hard if there's language barriers because, I only speak English and Spanish. Melissa speaks Chinese and English. Like we're limited. And then we have lots of community members who help with translation. We have folks in Germany and France and Italy, Brazil, so to speak Portuguese. But you need that person who's bilingual so to help bridge the gap there.
Yeah. Yeah. I mean, that's true. Like, I've thought about that too. Like, if Gilbert was not born here, like if we were not born where we had, the ability to genetic test him and we had the ability to stabilize him, like we would have never known because he probably would not have made it past 48 hours of life.
Yeah, and that's where we don't, we just, because again, genetic testing is it's not readily available in every country, right? And it's certainly not with maybe rapid turnaround.
Mm-hmm.
So I think that's certainly another goal, like newborn screening. Shelley, for many, many years was advocating for newborn screening of Barth and was told no, because there's no intervention, right? There's no drug that can be given. We still argue that knowing early is still very important, right? Because it will change how you monitor.
Right.
Right. You're going to be on the lookout for neutropenia. You're going to be on the lookout. You might be doing echoes, which you would never do in a, baby that doesn't seem to have any heart issues.
Yeah.
But if Elamipretide gets approved, that allows us to go back to advocating for newborn screening in a much more rigorous way, kind of meeting all the criteria for it. And then if we, succeed in getting that on the kind of newborn screening panels here, that helps other countries again advocate for their version of newborn screening to look for Barth as well.
That's really crazy. I didn't know that unless you have a treatment for something, they're not going to do newborn screening for it. That seems very.
That's one of the hallmarks. And I'm sure for some they do they do get on the newborn screening panel without having an intervention. But for Barth, that was what we were told. You also, the way that they test for it has to be a specific kind of test, although genetic testing is becoming so fast and the cost has come down literally by orders of magnitude that, I think it going forward, my guess is, in the next 10, 20, 30 years, they'll just be genotyping everybody at birth and looking for, then you're just going to try, you want to be on that list of things they're looking for.
Yeah. Did you ever see that movie Gattaca?
No, my husband
Oh my
loves
God,
that movie.
what?
I know. I've seen like no movies. That's what this comes down to.
Oh my God, Lindsay, someone with your background has got to have seen Gattaca. Number one, it's Ethan Hawke and Jude Law and Uma Thurman. And like it's 90s of those, so
Yeah.
they're like young and amazing. It's like I love that movie. Like, oh, okay, you need to see Gattaca because that is like literally what they do. Like it's in the future, and that's kind of like they're able to like screen and test and like
And
do everything in the beginning. And
I think there's
yeah,
lots of ethics discussions around this, and I'm not well versed in that. I will put that,
yeah,
but about, let's say they say, okay, you do have a mutation in TFASM, but we've never seen it before, so we don't know what it means. Like,
yeah,
and then the other thing that I've heard from the ethics perspective is like, you are making the decision for your child, and what if they would choose not to know? And I think, again, for conditions that affect you in infancy, I think it feels a little bit more clean cut. I think it's more stuff that's like oh, well, you might develop Alzheimer's, or this means you will develop it. Do you want to know that, but hopefully, like I said, I hope that Elamipretide will be, approved, and that then we can start really advocating hard again for Barth syndrome to be on newborn screening, so that you know in advance things to look for.
Yeah. No, I mean, I think that's kind of part of like our story is like we had a little bit of indication just because of like family history, which they
Right.
didn't know like that my brother
Right.
had Barth syndrome. They just knew it was heart failure. So we were a little bit cautious around like, is there going
Yeah.
to be something going on with his heart? But had we not known that and had known, nothing, I mean, we're constantly saying like, we're so glad that he was born, around the Christmas holiday and that we, the kids were with their grandparents because everyone was like, oh, you can like you guys you could discharge today if you want to, and we were like, no, it's okay, like we're gonna stay
Yeah.
home or we're gonna stay here. And like had we gone home it would have gotten very very scary very quickly because of how
Yeah.
quickly his
It just
started to
turned.
decline. Yeah, I mean,
Yeah.
so yeah, it would be nice to have newborn screening and be able
Yeah.
to catch things earlier because I think it's especially like in our community there's a lot of really scary things that happen to your baby and you don't know, you don't think anything was going on with them, and then to have
Right.
that happen is like it's terrifying. And it's
Yeah.
from what I've seen on Instagram of a lot of the other rare conditions and ultra rare conditions, like that is kind of what happens. You something really scary happens with your baby, and what if we
Yeah.
have the ability to kind of screen for some of those things initially?
Right. And just be monitoring more in real time rather than being reactionary, which is what you have to when you don't know. And with Barth, some people it is inherited, right? So mom carries a mutated copy of tafazin, and then there's a 50% chance that any boy will have Barth.
Mhm.
But it also happens what's called de novo, which means that there is no family history. And for whatever reason, in the egg that got fertilized, tafazin was mutated.
Mhm.
And so that baby ends up with Barth. And so you might not know you have, there may not be any family history. And, so newborn screening helps catch everybody.
Yeah, yeah. All right. Well, I feel like Gilbert's kind of gotten like at the end of his limit.
He looks very tired. He's like doing the eye rubs and the
Okay, so sorry about the abrupt ending, but as you may have guessed, Gilbert did decide that he was over it. So that's what happens when you co host with a 6, almost 7-month-old baby. I want to say thank you again to Lindsay for joining us today on In Rear Company. We will be back with more episodes. And if you want some more information about anything you heard today or would like to see more of the graphics and the images that we use, we will link those in the description. And again, thank you so much to Lindsay for joining us, and we'll see you next time.